Hello everyone, and good afternoon, and welcome to our very first webinar on proteomes. My name is Ramona, and I'm a curator with the Protein Function Content team at EMBL-EBI in Cambridge. Together with our international collaborators at the Swiss Institute of Bioinformatics and the Protein Information Resource in Washington, D.C., we define the content and representation of proteomes in the UniProt web interface. So UniProt is a comprehensive resource for protein sequence and functional information. The UniProt website acts as a portal to the different databases we provide. So previous webinars from the team have introduced to you some of these resources. You can access these through our YouTube channel, as well as through the EBI's training portal. Today, I'd like to focus on proteomes, what proteomes are, where the data comes from, and how to find the most relevant set of protein records for your work. So what is a proteome? A concise definition of a proteome is simply the set of proteins thought to be expressed by an organism. At UniProt, we aim to provide proteomes for every species with completely sequenced and annotated genomes. So I thought a simple schematic might be useful here. Let's consider the case of a small genome made up of two components, chromosome A and chromosome B. Raw data from a sequencing study might look like this, a set of short reads that overlap and therefore can be assembled into contiguous sequences or contigs. Further assembly of these contigs results in longer stretches of DNA called scaffolds, and this process is continued until entire chromosomes are assembled. This is followed by a process known as genome annotation, which uses gene prediction algorithms to identify putative coding regions in the genome. These coding sequences, or CDSs in short, are identified by a process, and this may be done either by the laboratory that sequenced the genome or by an independent group such as Ensemble. Coding sequences can then be translated into protein sequences. So a set of protein sequences arrived at in this manner makes up the proteome of the organism. So I just wanted to mention that genome assembly in this manner is time-consuming, and consequently we predict genes using gene annotation onto partially assembled genomes, which may consist of scaffolds or even just contigs. The set of protein sequences derived from such an assembly is made up of a single component, a master record that contains all of these sequences. Proteomes are updated approximately every 4 weeks, and updates to partial assemblies are captured in this manner. So next I want to talk about where proteomes come from. Just before each UniProt release, we have import programs that look for new sequence data and updated data from a number of different nucleotide sequence databases. The main databases such as EMBL, GenBank, and DDBJ are part of the International Nucleotide Sequence Database Collaboration, or the INSDC. The INSDC also includes a genome assembly database, which helps to group together sets of sequences corresponding to a single assembly of a sequenced genome. The genome size and genome coverage measures are taken into account by a procedure we maintain at UniProt to assess the completeness of each genome before including it in the proteomes database. The sequences imported from the different nucleotide databases are brought into UniParc, the UniProt archive, and this information is used to assess whether an incoming proteome is redundant to existing proteomes before including it in UniProt KB. Proteome-related information, such as the mapping to genomic components, is added to each UniProt KB record. The protein records that pass both of these filters, the complete proteome filter and the redundant proteome filter, make their way into the UniProt knowledge base. So going back to the UniProt web interface, we see that in the current version of UniProt, UniProt release 2016-05, we have nearly 50,000 proteomes spanning all 4 superkingdoms: eukaryota, bacteria, archaea, and viruses. Now, it is possible to browse the entire set of complete proteomes and to navigate your way to a proteome of interest using the search filters available on the left. But if your query is more specific, say you want the protein set for a particular species, you can use either a scientific name or a taxon identifier if available. So a simple search for example, for Bacillus subtilis or the taxon identifier 1423 returns a list of 70 proteomes. I've taken the example of a bacterium, as for many bacterial species, we now have several proteomes to choose from. For some other bacterial species, such as those involved in infection outbreaks, for example, Staphylococcus aureus, thousands of isolates of the same organism have been sequenced and submitted. So let's take the example of Staph aureus. This has more than 5,000 proteomes in UniProt at the moment. So for cases like these, it became imperative to introduce features to deal with this level of redundancy. So I want to introduce 3 features that we put in place over the last year and a half to deal with the vastly increased submission of genomes. The first of these is a set of reference proteomes. So reference proteomes are well-studied model organisms and other species of interest for biomedical research and phylogenetic studies. There are ongoing efforts to systematically choose 1 or more reference proteomes for each taxonomic group, and we do this using clustering methods and followed by curator verification. The reference proteomes are usually the best annotated proteomes for a particular taxonomic group. For larger taxonomic groups, you can have more than 1 reference proteome. At UniProt, we also want this set of reference proteomes over the entire taxonomic range to serve as a representative cross-section of the taxonomic diversity we see within UniProt. And finally, we want this set of reference proteomes to be consistent with NCBI's set of reference genomes. So let's take the example of one of these proteomes. Each proteome has a unique identifier that can be used to retrieve the set of sequences corresponding to that genome. So these sequences, 2,580 sequences in this case, can either be viewed on the UniProt KB interface, or the full set of sequences can be downloaded from the Proteomes interface in various formats. We also provide links to the genomic components from which these are derived. In this example, a chromosome and a plasmid. Proteomes are also associated with a genome assembly, 1 proteome for each genome assembly, and version changes to these genome assemblies and other updates are captured every 4 weeks with each new UniProt release. And finally, we also provide a short description of the organism sequenced and details of the sequencing project where available. The second feature I wanted to point out is the set of redundant proteomes. In April of last year, we developed and implemented a procedure to identify highly redundant proteomes within species groups. This was implemented for bacterial species where we see the greatest increase in exponential growth. And again, last year, this pipeline was used to filter highly redundant proteomes from the UniProt knowledge base. And following that, with every new release, this pipeline is used to filter the new proteomes coming in, and this has led to a more manageable rate of growth. So if we look at one of these redundant proteomes, the set of protein records corresponding to a redundant proteome are no longer present in UniProt KB, but these sequences are still available for download in the UniProt archive. For each redundant proteome, you will be directed to an alternate non-redundant proteome available for the same species. Let's go back and look at one of the reference proteomes. The third and final feature I wanted to introduce here, which we did to help users make the best use of proteome data, are pan-proteomes. Pan-proteomes provide a representative set of all the sequences within the taxonomic group. So these pan-proteomes are meant to supplement the reference proteomes by capturing unique sequences which are not found in a group's reference proteome. Each non-redundant proteome forms part of a pan-proteome within its taxonomic group. The set of pan-proteomes is intended to serve users interested in evolutionary studies, phylogenetic comparisons, and proteome diversity. I must also mention that it is possible to query proteomes using the proteome identifier as well as the genome assembly identifier, both of which are unique to each proteome. Here are some links to accessing UniProt data. And as I mentioned at the beginning, you can access this and other videos from the UniProt team, both at the EBI training website and the YouTube channel. And for questions, please use the chat box at the bottom of the Go Training box. Hello, hi. This is to answer the question about redundant proteomes. So basically, the redundancy minimization procedure was introduced to because there were lots of proteomes submitted which were highly redundant to existing proteomes. No proteome was actually 100% redundant to another, but since the majority of proteins was redundant, we had to find a way of keeping these out of UniProt KB. So we developed a method to do a pairwise comparison of proteomes within species groups and to identify those proteomes that are highly redundant, and by that we mean 95% redundant to other proteomes that already exist in these groups. And this was done to minimize, this was done to remove proteomes, but at the same time minimizing the loss of information. So we identified highly redundant proteomes and retired them, so the sequences exist in UniPark, but only the non-redundant proteomes, which is quite a small fraction of proteomes available for each bacterial species, remain in UniProt KB. Okay, the question about reference proteomes. So ideally, in the ideal situation, we would have only one reference proteome for each species group. But this is not always feasible because two different groups of researchers may have spent time annotating two different genomes, and therefore we cannot choose one over the other. In such cases, we keep both, or in the cases of very large species groups, we keep 3 or 4 proteomes. So it's never more than a small number. So you can choose the one that's best for your work, depending on, say, the number of annotated proteins in each proteome. So if you go to the species page for a set of proteomes, you have one of the things you can choose to display is the protein count. So you can look at how many proteins there are, and for each of them you can look at how many SwissProt records are available. So SwissProt is the expertly reviewed protein records, and if a proteome contains more than another, it will automatically be listed on top. So we have what we call an annotation score. How well annotated a proteome is reflected in this annotation score, and this annotation score is used for ranking the proteome. So you would see the best one on top, but you may choose to use a different one because it has always been used by your research community. So it's something you will have to choose. So I'm just looking at the question about finding the proteome of a bacterial species that is resistant to an antibiotic. So I imagine, although I've never done this myself, that species which are resistant to the antibiotic will express that protein, the resistance protein, and the others wouldn't. So you would have to do a preliminary search in UniProt KB and find the species of interest, and then from the taxonomy page you can link back to the proteome. So if you find the right strain, then from the taxonomy page you should be able to navigate your way to the proteome. The proteome construction. Yes, I can do that. Right, okay, so I wanted to explain how we arrive at the set of protein records making up the proteome from the genome. So when you do sequencing, basically what you're doing is fragmenting the chromosome up, and this is what is obtained in the form of reads. So what we want to do is to assemble it back and arrive at the chromosome, which is a long stretch of DNA. So this is what people do in sequencing. So reads are assembled into contigs, contigs are gathered together into scaffolds, and in the final stage of assembly, you would be able to get fully assembled chromosomes. But what I wanted to stress upon here was that this assembly process does not have to be complete for a proteome to be available. So with the advent of whole genome sequencing and next generation sequencing technologies, it is possible to just do the preliminary assembly to get contiguous sequences that are long enough to do a gene prediction on, and then you can arrive at the coding sequences. They may be fragmented, they may not be complete, and you may miss some proteins, but by and large you have the protein, the proteome available. So such a proteome would still pass the complete proteome test because the number of coding sequences would be comparable to its taxonomic neighbors. So this is to answer the question about pan-proteomes. So I guess it would depend on what set of proteomes you're interested in, but we cannot generate all of the possible pan-proteomes on the fly. So what we've decided is to limit ourselves to the pan-proteomes that users most often require, which is this a proteome that spans the entire species group. So the current pan-proteomes are limited to species groups. They do not include proteomes that belong to neighboring species. These pan-proteomes are constructed using the UniRef clusters, if you're familiar with them. They are not strictly taxonomic, they're not strictly species groups. So if within a species group you have two distinct sets of genomes resulting in proteomes, they would cluster independently, and so you would have a pan-proteome for each cluster. So it's an automated method, the clustering. So we cannot guarantee that it's always for a species group, but by and large, the clusters relate to species groups. You can read more about the pan-proteome or redundant proteomes or anything else in the documentation we have on the website. There is extensive documentation on the redundancy minimization that we carried out last year.